Today I'm going to talk about this title, but your main title is like this. So, exploring new horizon, novel microbial platforms for protein and biochemical productions. So, I'm going to talk about this plant secondary metabolite engineering in fusion is uh, POMBE. So maybe most of you are very familiar with Ogatae metanolica something, but I like to talk about the Pombi today. Not sorry, Saccharomyces and also Ogatae. And this is, I just want to introduce myself. So I belong to this International Center for Biotechnology, uh, in short, IC Biotech, Osaka University. So this is our building. And here is Osaka in Japan. So this is Osaka, and maybe if you come to Japan, maybe you can land at the Kansai International Airport. And our campus is here, Sita campus. And our International Center for Biotechnology has some missions. So our main research project, international cooperation among Asian countries, particularly ASEAN countries. So that's why we have many friends from the ASEAN people here. And we do some research and educational advancement in the field of biotechnology. And our main project are development and the utilization of biological functions, optimization of bioconversion processes, and conservation of genetic resources. And also, I would like to emphasize human resource development, I mean the student educations. And this is human resource development. So I have some programs. So if Kent or Indonesia or Vietnam professors are interested in, please just tell me. So we have a graduate school of engineering. This is English course. All lectures are given in English, and frontier lab at Osaka University. So your student come over to Osaka, then try your research at Osaka University and double degree programs. And also nowadays Osaka University focus on two projects. One is ASEAN Campus Project. So Osaka University International Certificate Program, the OICP, this is graduate students. And I just give the name, OU ASEAN Biotechnology School. This is undergraduate student programs. And we also have a global knowledge partners, but IC Biotechs together works together with Aachen University and Bill Health and UC Davis. I'm sorry, we do not have a MOU with Kento University. So if you have a chance, we are very happy to make a MOU with Kento University. And this is our research station in Thailand. So we have a research station in Mahidol University since 2002. And this is our research facility, very tiny facilities, but we can do some experiments in these facilities. Then this is my town. So if you could, you had the chance to visit Osaka University, sorry, Osaka, maybe you know. So this is our foods. And Osaka is very famous for Japan's soul kitchens. So if you like to survive in Japan, in Osaka, the food should be cheap and good. If not, the shop will fade out. Okay, so I'd like to focus on my research topics. So I'm interested in flavonoids. So flavonoid is, as you know, a large family of plant secondary metabolites and have a biological benefits and exist as a glycoside forms. So you can see this is a glycans and this is glycan. Glycan part can contribute enhanced solubility, stability, or alter the biological properties. And glycoside engineering, I like to show one example. So this is hesperagine and starch. Then using cyclodextrin glucanotransferase, so we can add a third glucose moiety here. Then increase this molecule get increase solubility and absorbed more rapidly. Now this molecule is used as a one ingredient in cosmetics. So my today's presentation objective is create flavonoid derivatives using fission yeast. So this is our idea. So we can give the flavonoid molecule, sorry. Yeah, we can give the flavonoid molecule to the fission yeast, then this molecule go into the cells, then using UDP sugar and glycosyltransferase, we can make a new flavonoid molecules. And in our research, we focus on the citrus flavonoids, so naringenin or hesperagine. And this molecule is considered as reduction, has a function of reduction of blood pressure, improvement of circulation, and anti-cancer activities. So in my presentations, I like to focus on this citrus maxima and also orange citrus sciences. And this orange is a gene source of alpha-1,2 transferase, and this orange is a source of alpha-1,6 transferase gene. And this is our idea. This is the laminosylations in citrus fruits. So here is, thank you. So this is the flavonoid 7 glycoside, and we can use the UDP laminose to give the laminose residues to these flavonoid molecules. We use two enzymes, 1,2 laminosyltransferase and alpha-1,6 laminosyltransferase. Then this enzyme can transfer a glucose, laminose is here, and this enzyme transfer the laminose is here. But just the linkage is different, then the property can be changed. This is a bitter taste, and this is a tasteless. Just one glucose molecule, we can change the property of biological molecule. So we are interested in this point, so we try to change the flavonoids sugar chain structure using the different enzymes. So the problem is we do in the East, they do not have, UDP laminose is not commercially available. So we have to synthesize this UDP laminose in vitro. So we try to express this UDP laminose synthetic enzymes in the East. So this is an experimental skill. I'm going to talk about three parts: UDP laminose production in fission yeast, and the characterization of citrus laminosyltransferase enzyme substrate specificity, and also we try to produce the flavonoid derivatives in vivo. So this is the UDP laminose production in fission yeast. Why we are interested in this schizo Saccharomyces pombe, because I recruited Dr. Ohashi from this Asahi Grass company. So this Asahi Grass company developed this recombinant protein production systems. So he joined our research group with this technology. And using these systems, we try to first synthesize these UDP laminose molecules. Then we introduce this gene, Arabidopsis gene, into the fission yeast, and we extracted the nucleotide sugars and analyzed by HPLC in vitro. And here is the SDGs page. I mean, recombinant enzyme expression, how we confirm the protein productions using SDGs and Western blotting. And you can see new peak here. This peak correspond to the UDP laminose. Now we can produce the UDP laminose in a fusion yeast. Then using this UDP laminose, we analyze more enzyme activities. So the second is characterization of citrus aminoacyltransferase. So we use this alpha 1 2 aminoacyltransferase, and we express to 1,6 aminoacyltransferase with a histidine tag. Then we can detect this enzyme expression in fusion yeast using Western blotting. Then we analyze the in vitro activities using these systems. So we give the UDP laminose, and we use these kind of flavonoids molecules and analyze using HPLC. Then here is the data. So this is the acceptor molecules we used. Then you can see new peaks here. So A, but B and C eluted at different positions from the position A. But the mass spectrometry data is the same. So we further analyze this molecule, then identify. So molecule A is naringin, and molecule B, narilgin. So the product's structure is different. Then here is a summary of our research. So using this N7G as acceptor, Q3G used as the acceptor, and this is the product. So when we use alpha 1 2 aminoacyltransferase, this enzyme can produce only naringin. But in the case of alpha 1,6 aminoacyltransferase, they can produce naringin, but they can produce more narilgin. So substrate specificity is different, but this alpha 1,6 aminoacyltransferase can also transfer aminoacyl molecule here and here. So we could say this enzyme's substrate specificity is very broad. So now I'd like to move on to the development of in vivo glycosylation system. So here is schizosaccharomyces pombi. Then we give the N7G as the acceptor molecule. Then this molecule go into the cells. Then this UDP laminosyl synthetic enzyme works, and this using these products, then this transferase enzyme can transfer to the laminose residues to the naringin. N7G, then we may be able to get naringin in vitro. So this is a concept, and we did experiments. And here is, we just confirm our recombinant protein enzymes are produced in a fusion yeast. Then we analyze our enzyme reaction products done in the cells. So here is, we just give the, we just give the N7G as acceptor molecule in a cultivated media, then is incorporate this molecule, then recombinant enzymes transfer the laminose residues to this molecule, then we could get the naringin molecule in the yeast cells. So N7G was laminosylated in fusion yeast. So now we can produce laminosylated N7G using Pseudosaccharomyces pombi. So I just want to show you the another recombinant protein host not only Saccharomyces, but also not only Ogatae methanolica. Maybe you can use this system to produce your recombinant proteins. Okay, so this is a summary. Conclusion: in vitro activity of citrus aminoacyltransferase was detected. Then we can produce the naringin molecule in fusion yeast. Okay, so I would like to finish my talk, this one, but I would like to add more informations. So since yesterday you talked about biofuel productions, so we also try to use Rhodotellula toluloides, and this is very robust yeast cells. Then we can use these yeast cells. This is the Basidium mycetus oleaginous yeast. Then using these cells, we can produce the plenty of fatty acid. Now, what we are doing is we use the waste glycerols. So maybe in Thailand, in Japan, cooking oil or restaurant waste oil is harvested. Then some company harvests all the waste oil. Then they produce fatty acid from waste oil, cooking oil. Then plenty of waste glycerol is produced. So we like to use this plenty of waste glycerol as a carbon source or to the grow this Rhodotellula toluloides. Then we succeeded these experiments, and we can produce plenty of fatty acid using waste glycerol. Then someone talked about the stress tolerant Saccharomyces cerevisiae. So in our case, to reduce the cost for during the fermentations, we try to isolate the thermo tolerant mutants, Rhodotellula toluloides. Then originally these strains can grow at 30 degrees. This strain is isolated in Thailand, but this strain cannot grow at more than 30 degrees. So we tried to isolate the thermotolerant mutants. Then finally, these thermotolerant mutants grow at 37 degrees. Then we analyze the properties of these thermotolerant mutants. Then this mutant shows ethanol and hydrogen peroxide stress tolerance. And I would like to show you another example. We have a research collaboration with Biotech, Dr. Nilan and Dr. Berawat. So we have some publication use this Ogatae methanolica. And I also work on, sorry, this from here, this is beyond microbes. Very sorry to talk about this one, but we also try to produce the IgG using Nicotiana bentamiana plants. So we successfully produce this Barilumab antibody in tobacco plants. And I would like to show you another example. So glycoengineering, we are very much interested in. So someone introduced the glycosylation reduce. I'm very much interested in that phenomena. So in our case, we create glycoengineered plants, glycoengineered silkworms. Sometimes we try to produce glycoengineered yeast for the recombinant protein production, such as vaccine or pharmaceutical proteins, something. So this is our data. So she is Thai, and she did this kind of experiments in our laboratory. So she successfully expressed glucocerebrosidase enzymes in Nicotiana benzamiana. Then we analyze sugar chain structure like this. Then here is another example. So this is Kaiko. Kaiko is a venture company, but the Kaiko means a silkworm in Japanese. So we can express recombinant proteins using silkworm. And this venture company tried to produce porcines sarcovirus type 2. So yesterday someone mentioned these vaccines, but they can produce these vaccine proteins in silkworm. Then they try to test this vaccine in Vietnam. So I hope we can apply silkworm mates the vaccines in Vietnam and also maybe in Thailand if you want. So maybe they can make another clinical trials in Vietnam or in Thailand too. Okay, so thank you very much for your attention, and I hope you could get some informations from my talk. Thank you very much.
Thank you, Professor Fujiyama, for impactful presentation. And for the session, Q&A session, we still have 9 minutes left. We have time for questions and answer. And if anyone has a question, please raise your hand and a member of our staff will bring you a microphone. And those who are attending online, please feel free to type your questions in the chat box. Dr. Piyanan will read your question out loud for the presenters. Question, please.
Thank you for your talk. I wonder if you produce the recombinant protein in plants, so that the step would be easier to use in human than in microbes, because I'm not familiar with plant at all. Can you maybe elaborate a little bit? Thank you.
Okay, thank you for your questions. So nowadays we can just need 1 week. So we use the transient expressions. Previously, we prepare recombinant agrobacterium, which carries the plasmid. Then we have to transfer the gene to the plants. Then we have to regenerate transgenic plants. So it takes about 1 year at least. But now, once you prepare plenty of plants like this, then we can give the agrobacterium. Then next week, you can harvest plenty of plants. So our experiences, just 1 leaf of the plant can give you about 300 microgram of IgG molecules.
Hi, thank you for your talk. I wonder if you can comment on the secretion potential of POMB compared to good secretor, like for example, Pastores or something.
Thank you very much. So far, honestly speaking, we try to produce the flavonoid molecules in the pseudosaccharomyces pombe. So I have not studied how our recombinant protein go into the media, secrete into the media. But according to the publications, the secretion mechanism is very similar to the Saccharomyces cerevisiae. So maybe depending on the proteins, the efficiency would be changed, would be different. But I believe Schizosaccharomyces pombe can also secrete the protein like a Saccharomyces cerevisiae.
Thank you.
Hello. Great talk. I have a few questions, but I'll limit myself to one. With the silkworm research, where do the proteins localize? Like when you're producing them, can you control where you're localizing them? Yeah, how much control over the organism do you have?
So in this case, the silkworm can produce the proteins, the full body.
Okay.
So the glycosylation pattern is different.
Mm-hmm.
But if you like to focus on the humanized sugar chain structure, we should produce the recombinant proteins in middle glands.
Okay.
So middle ground means they can produce the silk proteins.
Okay, that's interesting. Because the only reason I was asking is like, I know that silkworms are able to like eat plastics anyway. So if you could get a protein being produced in the gut that was like a PET hydrolase, you could have like a double system of decomposing these types of plastics. And
Yeah.
it's something we've looked at previously.
So the vaccine utilizations, they should produce the silkworm inside, like a pupa or something,
Okay, yeah,
or larva.
nice.
But once they produce the recombinant proteins on the cocoon, maybe it's a bit different story.
Yeah, yeah, perfect. Thank you.
Hi, thank you for the talk. I have a question about the fission yeast story. I noticed that you feed the N7G to your yeast. Do you have any? Do you plan to engineer the yeast to for the production, de novo production of N7G, so you don't have to feed N7G into the system? Is it possible? Do you have the tools available to engineer the yeast to produce it in vivo? Thank you.
Yeah, thank you very much. Interesting questions. We have not tried the such synthetic biology, but in the case of Saccharomyces cerevisiae, as you know, they can produce the flavonoid molecules in the Saccharomyces cerevisiae by introducing many genes into Saccharomyces. So I believe we can do all the same trials in Schizosaccharomyces sponbi, but we haven't tried that experiment yet.